Here we have an assessment standard question based on the content of our last lesson. So we've got a diagram that represents the clonal selection in lymphocytes. You're being asked to identify what stimulates the division of cell Y. The division of cell Y is stimulated by the binding of antigens to receptors on the cell membrane. The antigen, remember, is on the pathogen, and the receptor is on the surface of the lymphocyte. In this PowerPoint, we have two learning intentions that we are covering. The learning intentions are looking at the action of T lymphocytes. So by the end of this PowerPoint, our aim is to be able to describe the action of T lymphocytes with regards to production of antibodies and their role in defence. And we want to be able to identify or explain what is meant by an autoimmune disease. In our previous learning, we have been looking at lymphocytes, which make up the specific immune response. Lymphocytes are derived from stem cells in bone marrow. We have two divisions of lymphocytes, B lymphocytes, which we looked at in the last key area, and the production of antibodies, and today we're going to be looking at T lymphocytes or T cells. Again, as a recap from our last lesson, lymphocytes are white blood cells that are involved in the specific immune response. T lymphocytes destroy infected body tissues. They recognise the foreign antigens of the pathogens which are displayed on the membranes of cells that they have infected. T lymphocytes are therefore described as being cell-mediated. That means that they attack our own body cells after they have become infected by a pathogen. The action of T lymphocytes is important because not only do they kill infected cells, they can also further stimulate an immune response. So T lymphocytes, as we've already identified, can recognise foreign antigens on the surface of membranes of our own body cells. T lymphocytes are then able to bind to these antigens specifically, and they release proteins into the infected cell. These proteins cause the cell to produce self-destructive enzymes which cause a programmed cell death called apoptosis. The action of the T lymphocytes then attract phagocytes to the area where the remains of the infected tissue can be removed by the process of phagocytosis. There are a variety of T cells within our body, and they bear antigen receptors, just like our B lymphocytes did, that are able to recognise and bind with a foreign antigen on the surface of what we call an antigen-presenting cell. When we get this antigen recognition, T cells become activated, and what they will form is either a clone of activated T cells for immediate use, or a clone of memory T cells for future use if necessary. The memory cells we're going to look at in our next PowerPoint. The activated T cells move towards a site of infection and will attack the infected cells. T cells release proteins which diffuse into the infected cells and induce them to produce self-destructive enzymes. These break down the cell's DNA and vital proteins, causing the cell to die. This process of programmed cell death is called apoptosis. From the diagram in front of us, we can see at the top that some T cells have been activated as a result of a pathogen that has been detected via the antigen recognition system. The T cells, after being activated, have moved towards the site of infection, moved towards the infected cell, and they attach onto the infected cell using the receptor antigen binding. The T cells release proteins which diffuse into the infected cells, which we can see down at the bottom, and induce them to produce self-destructive enzymes, which is represented by the little red one right down at the very bottom left-hand corner. These self-destructive enzymes bring about the cell's DNA and vital proteins being broken down and causes the cell to die. This process is called apoptosis. As a result of apoptosis and the action of the T lymphocytes, we can see that a phagocyte is also attracted towards this site, and the remains of the destroyed cell will be engulfed and destroyed under the action of phagocytosis. T lymphocytes attach onto infected cells and release proteins. These proteins diffuse into the infected cells, causing production of self-destructive enzymes which cause cell death. The remains of the cell are then removed by the process of phagocytosis. Each person's body cells are different because they possess a combination of cell surface proteins known as their antigen signature. This antigen signature is unique to every individual. It is of critical importance that a person's lymphocytes do not regard that person's own body cell's surface proteins as antigens and attack them. Normally this does not happen because any lymphocytes bearing an antigen receptor that would fit a body cell surface protein is weeded out and rendered non-functional or destroyed by apoptosis. Surviving viable T lymphocytes are able to distinguish between self-antigens on the surface of the body's own cells and non-self antigens on the surface of infected cells or cells not belonging to the body. This ability to recognise self and non-self means that normally there are no lymphocytes acting against the proteins on the surface of self body cells. Sometimes, however, the body no longer tolerates the antigens that make up the self message on cell surfaces. Such a failure in the regulation of the immune system leads to T lymphocytes launching an attack on the body's own cells. This process is called autoimmunity, and it is the cause of autoimmune diseases. Some examples of autoimmune diseases include type 1 diabetes and rheumatoid arthritis. So for our notes, in autoimmunity, the T lymphocytes attack the body's own cells. This causes autoimmune diseases such as type 1 diabetes and rheumatoid arthritis. Here we have two assessment standard questions focusing on T lymphocytes. The first is a level C, the second is a level A. Take your time to read through the questions and think about the answers. To explain how a lymphocyte is able to recognise a particular pathogen, we would be looking here for that it recognises the antigen or it recognises the specific antigen on the surface of the pathogen. to describe how T lymphocytes destroy infected body cells, they cause the cells to produce self-destructive enzymes, or they induce what we call apoptosis or cell death. At the end of the PowerPoint, we're asking you to reflect upon your learning, or the two learning intentions shown in front of you, and if there's anything that you're unsure of, please go back to the appropriate slide and familiarise yourself with the content again.